Amidst the ravaging COVID-19 pandemic, you're bound to have heard the word antibody or vaccine before. And if you haven't, well, pause this video and go look at the news.
Antibodies.
Antibody test. An antibody.
Development of vaccine. Coronavirus vaccine.
Vaccine.
Now, what exactly are vaccines, and what is the sciencey work that goes on behind the laboratories making them? And keep in mind that antigens are anything that can trigger your body's immune response, and that antibodies fight against antigens. Vaccines initiate the primary immune response by stimulating your body to create antibodies against a specific antigen. So the next time the virus comes around, your immune system is prepared with an antibody army. But how is this achieved? Is it all mystery stuff? Nope. The good old-fashioned vaccine contains a dead or weakened virus, or just antigen parts of the virus. When injected in humans, this triggers the primary immune response and subsequently the formation of antibodies. But wait a minute, creating vaccines isn't that simple. The most common vaccine-making procedure is an egg-based vaccination process. Scientists inject dead or weakened viruses into a chicken egg. Yes, a chicken egg, the same type of egg you ate this morning for breakfast. Injecting the attenuated virus into an egg not only allows weakened viruses to proliferate, but also allows more time for the virus to become mutated and less efficient at infecting humans. Scientists then purify the pathogens and voila, the vaccine is ready. But as you can imagine, this process requires mass amounts of eggs, and one egg only generates one vaccine. It would take years to produce a COVID-19 vaccine, test it, and distribute it to the people. And who wants to stay in lockdown for that long? Alas, a newer, more modern way of vaccine making has come around. This futuristic vaccine is known as an RNA vaccine. How exactly is an RNA vaccine made? Let's use the novel COVID-19 as an example. The main antigen of the COVID-19 virus is a surface spike protein. On January 12, 2020, the entire COVID-19 virus genome was sequenced. Scientists identified the amino acid sequence that coded for the COVID-19 spike protein and began to replicate its messenger RNA. The messenger RNA could be translated in the ribosome of any cell, be it a mouse or human, to produce spike proteins identical to those of the COVID-19 virus. Scientists then injected the messenger RNA into an organism. And why would they do so? Cells that uptook the RNA began to form COVID-19 spike proteins, which acted as antigens and triggered the formation of antibodies in the cells against them, rendering immunity. That exposure and antibody formation acted as a primary immune response against COVID-19 spike protein, and the organism would be prepared with specific antibodies and memory B cells if the real virus attacked. Then why isn't the vaccine all over the world now? Why are people still dying, countries still affected, nations still in lockdown? Well, vaccines are distributed to around 3.1 billion people per year. There are 8 phases to developing vaccine and an entire process for approving it. Safety of the public and disease enhancement are 2 major reasons it is necessary for scientists to test a vaccine and make sure it is not harmful. But remember, as viruses keep evolving, so do we, and the race against viruses is on.